Yeah. Yeah, we're live now.
Yes.
And there are more people joining the
YouTube?
No, the Zoom session.
Ah, okay.
Okay, I think I'm going to disconnect my microphone, and Jessica, you start at 11.
Oh. Sorry, had to close the window.
Because there was a noise, a plane or something. Yeah.
Okay, so Noemi, I will start and I will present you. And in the meantime, I think people will connect, and then you can start your talk. So, okay, hello everybody. Thank you very much for being here today for this new webinar of the IGCP 777 project. For those who join us today for the first time, I would like to mention that this project has the aim of better understand the origin, distribution, and biogeochemistry of arsenic in the Altiplano Puna, and is funded by the International Geoscience Program of UNESCO. The leaders of the project are Jocelyn Tapia from Universidad Católica del Norte in Antofagasta, Chile, Mauricio Ormachea and Noemí Tirado from Universidad Mayor de San Andrés in La Paz, Bolivia, Kirk Nordstrom from the USGS Boulder, Colorado, and me from CONICET and Universidad Nacional de Salta, Argentina. During the previous seminars, we were mainly focused on the hydrogeochemistry and origin of arsenic, not only in the Altiplano Puna, but we were also learning about those environments and chemical conditions on planet Earth that favor the presence of arsenic in waters. Today, we are going to focus on the effects of arsenic in human health and how some communities have developed an efficient arsenic metabolism. Today, our speaker is Noemí Tirado Bustillos. She is an emeritus researcher and professor at the Universidad Mayor de San Andrés in La Paz, Bolivia. She is the head of the genotoxicology unit and director of the Genetics Institute in the medicine faculty of that university. Noemí is the vice president of the executive committee of the Latin American Association of Mutagenesis, Carcinogenesis, and Environmental Teratogenesis. She is also the president of this society in Bolivia. She's a full member of the Organization for Women in Science for the Developing World. Noemí is coordinating and leading different research projects on environmental toxicology and genotoxicology related to health effects from exposure to pesticides, heavy metals, and other environmental contaminants. She was invited speaker in different national and international conferences and received several national and international awards in the area of genotoxicology. So thank you very much, Noemí, for your talk today.
Okay. Thank you, Jessica, for the presentation. And well, I would like to thanks to all the people that are following our webinars. So in this, all the attendance. In this time, I'm going to talk about the arsenic exposure, metabolism, and genotoxicity among women in Bolivia. So as a first of all, I would like to have this background about the arsenic in the world. Arsenic affect worldwide and more or less 2 million people and distributed in more or less 70 countries. In our continent, in Latin America, 14 of 20 countries are affected by the high concentrations of arsenic. So more than 10 million people are estimated to be exposed to arsenic. So in this map, you can see the distribution of this arsenic in the water, drinking water in different countries, Chile, Argentina, Uruguay, Peru, and a small part of in Brazil. The chronic environmental disease due to this water contaminated with arsenic, salts of arsenic, is described in Bangladesh, for example, and there are some other reports in Chile, Taiwan, Thailand, in Mexico, and Argentina. The Environmental Protection Agency have these maximum contaminated levels of arsenic in drinking water, that is 10 ppbs or 10 microgram per liter. As you can see here, a typical sign of this intoxication by arsenic is in the plant of the hand and in the food also. Well, talking about the chronic arsenic exposure adverse health effects in different systems of the people, we can see that the metal can affect different systems. In the nervous system, for example, we can find some impaired intellectual function, impaired motor function, heart diseases, hypertension, heart attack, and coronary heart disease. Renal system, there are described some kidney cancer or bladder cancer also. In the skin, these typical skin lesions that also can conduce to a skin cancer. The endocrine system will be affected as diabetes. We can find cases of diabetes, impaired glucose tolerance in pregnant women. In the respiratory system, there are described pulmonary tuberculosis, bronchiectasis, lung cancer also. And in this organ, the liver, we saw some descriptions of liver cancer too. In the kids, there are some developmental process affected. For example, increased cancer risk as the children become adults. Increased infant mortality, neurological impairment, and reduced birth weight on these children. Well, talking about the absorption, metabolism, and elimination of this metal, the arsenic came into the body by different ways, could be ingestion or inhalation. But in this case, we're talking about the ingestion in the water contaminated. It came as arsenic pentavalent. So it conflues to the blood and goes distributed to different organs, the liver, kidneys, muscles, bones, and we can find it accumulated in the skin or in the hair. The arsenic pentavalent and arsenic becomes as 2 arsenic trivalent or arsenic 3. This arsenic trivalent binds to a sulfur hydroxide molecule and could interfere with the cellular respiration and also with the glutathione metabolism and to the DNA repair pathways. And we can see the persistence of this molecule in hair and in nails also. This molecule, inorganic molecule, suffer methylations. So we can find the dimethyl arsenic acid, or DMA, and monomethyl arsenic acid, MMA, in the urine. The half-time is the 10 hours of these 2 demethylated and monomethylated arsenic. The organic arsenic that could be introduced to our organism will be eliminated by urine unchangeable or unchanged. In this schematic representation, we can see how this arsenic molecule could be biotransformed in our organism by the different metabolic pathways. This arsenic pentavalent suffer a conversion to arsenic trivalent, and this can convert to methyl arsenate or MMA pentavalent by the action of this enzyme arsenic 3-methyltransferase. And also there are acting this molecule. For example, there is adenosyl methionine that get reduced, and this molecule suffer another conversion to methyl arsenose MMA3. This molecule can convert to this dimethyl arsenic, so suffer another methylation by the interaction of a kind of arsenic 3-methyltransferase enzyme. So these two molecules that are inside of these red circles are the molecules that we can find in the urine. So if we can find the more concentration of MMA or more percentage of MMA in the urine, there is a toxic outcome. This is the molecule more toxic. And in the case of finding the more elevated concentration of DMA, that means that the person is efficient in the elimination of this compound, this arsenic, so that means effects are not going to be very toxic. So this is the main activity of efficient elimination of the arsenic is in this elevated concentration of DMA. Well, this metabolism, this conversion of the molecule of arsenic in 2 steps of methylation, it depends on the arsenic 3-methyltransferase genotype. So there are some variations of polymorphisms in the body that could follow the transformation, the methylation of this arsenic. So it could be converted in this optimal way of elimination, DMA. So these polymorphisms are different. For example, here we can describe the variation of RS 11, 19, 40, 39 in the C allele that could be could act in the first methylation, transforming the inorganic arsenic to MMA or monomethylated arsenic. In this other step for conversion of monomethylated arsenic to demethylated, we can see another molecule of polymorphism of arsenic 3-methyltransferase, the RS 3740 39-3, that could be the ways of the type of alleles that could allow the methylation of the inorganic arsenic in the body. Well, all that could affect these steps to convert the inorganic arsenic in a molecule that could be eliminated in a more efficient way of the body. Also, there are some other biological and environmental factors that could influence in the elimination and the transformation of this metal. For example, the age, the sex, arsenic exposure levels, of course, smoking habit, and nutritional status and diet also, that are potentially related to the inter-individual variations. Well, talking about the genotoxicity of this metal, the arsenic, could act or interact with the DNA, the desoxyribonucleic acid molecule, and could affect this molecule in different ways. For example, could cause aneuploidies, micronuclei formation, for example, as DNA damage, chromosomal aberrations, sister chromatid exchanges, and some others like cross-link DNA proteins. So this inorganic arsenic is classified as carcinogen class 1 in humans by the International Agency of Research in Cancer. The representation of this arsenic genotoxicity is in this schematic. For example, the arsenic pentavalent is introduced into the cells, and as I show you in the previous slides, it converted to arsenic trivalent. And due to this arsenic 3 methyltransferase enzyme could convert in monomethyl arsenic trivalent, monomethyl arsenic pentavalent, and dimethyl if it's going to be in the second step of methylation. So this molecule, the MMA, is one of the most toxic, so it could increase the genotoxicity. It means that it could increase the DNA damage. But in the other hand, we have the DMA that I show you. That means that if we can find this DMA pentavalent, the genotoxicity is going to be lower. But the genotoxicity of this arsenic molecule or arsenic metal is not directly interaction with the DNA. It forms or generates some reactive oxygen species that are shown here. So that means that the damage is going to be as oxidative production of oxidative stress. But also this reactive oxygen species could cause DNA damage or could increase the DNA damage, causing some double strand breaks, for example, in the DNA molecule, adducts or mutations, or also single strand breaks or deletions that are described here. And also that, as I told you before, the DNA damage of this reactive oxygen species, the arsenic produced, it can be also affect in the decreasing the optimal DNA repair system. Because if the DNA suffer a damage, could repair this damage by the system of base excision repair, for example, and non-enjoin repair. The mitochondrial dysfunction also is another affected organ by abnormal apoptosis signaling, for example. Well, the background of the arsenic in this way, we can see the place that we were taking in this study is located in the Lake Popo. As you listen in the previous webinars, we took these data of Ramos et al., for example, that found the median of concentration of this arsenic in the drinking water from 41 microgram per liter as medium, and the range was 5.6 to 242 microgram per liter. And this was described by Ramos et al. In 2012. And of course, the other studies done by Ormache et al. In 2013 that found more or less the same medium, 44 microgram per liter, and with the range of 5.2 to 207 microgram per liter. This is a geographic location of Lake Popo. It's located in Oruro department, and it's close to La Paz, more or less 200 meters of difference. So the study area of these studies was compounded by 10 villages around this lake. So we found these 10 villages, and as you can see in the different color of dots, there are the levels of the arsenic in the drinking water were from less than 10 microgram per liter to more than 200 microgram per liter. So we took these 10 villages, and the people recruited in this was of women exposed. So the evaluation of human exposure was done by the indirect evaluation. Water samples from wells and tap water were analyzed by a kit, a portable kit, to confirm that there was presence of this arsenic in these places. So we used a semi-quantitative kit, trademark Merck, and we also took some other samples to confirm the concentration, and we sent these samples to Karolinska Institutet to be analyzed and to have these concentrations. The participants, as I told you, were 201 women that were we applied a survey, and of course with the previous signed consent of these women. They were measured some physical parameters, for example, weight and height, and we took samples from blood and urine to perform this some analysis. Some pictures of these field works. We perform some quickly assays to give back to this population, for example, urine infections and hemoglobin and some other for example, the pressure and some other quickly assays that we gave them in the field. And then we took the samples, the blood samples and the urine samples to the laboratory to perform the analysis. And the urine samples and some blood samples were sent to Sweden to analyze the urine arsenic concentration and the metabolites also. Well, some results of this study, we can see the characteristic of the study woman around this Popo Lake. So we can see the differences between, for example, age. We took, we divided the population in two ethnic groups that there are in these villages. We found Aymara Quechua ethnicity and some few people or women from Uru ethnicity. So we found differences between age, for example, body mass index, and the concentration of urinary arsenic in microgram per liter. We found in the concentrations of total arsenic in urine. There were differences between these two groups, and also in the percentage of MMA and percentage of DMA also we found differences. But there is a surprise that we found that the people, the women that we took in this study had the of DMA. So that means that they were very efficient in eliminating this arsenic that is in the body. So that's why we couldn't find any sign of this affection or intoxication of the arsenic in all these people. Well, the arsenic exposure in these villages, here we can see in this graph a wide range of medium around these villages. So we can find, we found from 32 to 191 microgram per liter of this arsenic urinary. And also in some villages, for example, we found differences of a wide range of arsenic in this village, Cebarullo, from 28 to 630 microgram per liter. Well, performing a linear regression model, we found some significant results. For example, the urinary arsenic was not well associated with any arsenic metabolite. Higher body weight was associated with a lower percentage of MMA. The fish consumption we also analyzed because the arsenic could be intake or introduced to the body also in the consumption of fish. And the fish consumption was associated with a more efficient metabolism. Also, we analyzed the smoking habit, so it was associated with less efficient metabolism. And here. And then the influence on arsenic metabolism was seen by ethnicity. So the Uru population have more efficient elimination of the arsenic than the Aymara Quechua group. In summary, we can see that the woman had a wide variation of urinary arsenic. So the range was 12 to 407 microgram per liter. And as I told you before, there was a markedly efficient metabolism of arsenic. So we found a very low percentage of MMA in these populations. And in this graph is comparing to another populations, for example, in Argentina, the San Antonio de los Cobres, in different times, in 1995, in 2015. And here is the population that we analyze in Aymara Quechua and Uru. And as I told you, the Uru have the more efficient metabolism of arsenic, and this is most population, another population from other places. So in conclusions, the women studies in the community are highly exposed to arsenic in drinking water, which could constitute a major health risk. The women living in these villages around Lake Popo had elevated variant of inorganic arsenic exposure, and women presented low percentage of MMA and high percentage of DMA in urine. The ethnicity, body weight, fish consumption, and tobacco smoking were associated with arsenic metabolism efficiency. This is the first paper that was published in 2019 of this work that I present you. And quickly, I will show you some other results because this part is not yet published, and we are still performing some other complementary analysis. So in this case, the genotoxicity was analyzed including the 2,201 women, and we increased to 215 women that we call them exposed. And we took another population that has a similar characteristic of habits, living habits, and high altitude and altiplano, close to La Paz, that are non-exposed. So we perform, we used exposure biomarkers, these urine metabolites, arsenic, MMA and DMA, and effect biomarkers of DNA damage, that it's COMET assay, and the susceptibility biomarkers that we perform genetic polymorphisms of arsenic 3-methyltransferase in this RS 3740-39-3, performed by real-time PCR. So some results of these evaluations. In this case, we can describe the characteristic of this study woman, the cases and the controls, and these are the exposed women, and this is the control group that is in close to La Paz. This is the Popo lake population. So we see here that there are similar characteristics of these two populations, no differences, and also the high altitude. And we distributed these groups according to the median of those exposed from Lake Popo by the urinary arsenic concentration, because in the control group, we are waiting for these results of arsenic urinary concentrations. But so we took in this population exposed, we divided the population into groups, less than 60 microgram per liter of arsenic and more than 600 of, 60 microgram per liter as urinary concentration. So the arsenic metabolism according to the level of exposure is, as we saw in the previous study, the more percentage of DMA in this in the both groups, less than 60 and more than 60 microgram per liter. We found the efficiency of elimination of the arsenic. Talking about the urinary arsenic and DNA damage by COMET assay, this is the control population. It's 71 women, and here we have these exposed people or women that were divided in less than 60 and more than 60 microgram per liter of arsenic. And we found a DNA damage in the women that have more than 60 microgram per liter of arsenic in urine. And these are significant p-value. Then we divided these populations. This is the people, the women control, and these are the people or the women that are living in different places of the Popo Lake. This is the represent the people that are, or the women that are living in the south of this Popo Lake, and this is in the north. So here are described the villages in the north and in the south, and we found differences between the percentage of DNA damage. So the people that are living in the south, and also they have more concentration of arsenic in the urine, have more DNA damage than the other villages. So performing some correlation of the metabolism of and then the genetic damage, we performed this heat map, so we can see the p-value for Spearman test. We found some positive and negative correlations. For example, in the taking the tail moment, that is indicative of DNA damage related with the urinary arsenic. There is a positive correlation. Also, in taking into the percentage of DNA damage in tail with the DMA, percentage of DMA, and some negative correlations for the percentage of DMA and DNA percentage in tail. Also, the body mass index was show a negative correlation with the percentage of DMA. Well, the association of DNA in tail with arsenic metabolism, we can see in this table that we found some association of this the DNA damage correlated with the arsenic metabolism with the significant p-values in the, inorganic arsenic and percentage of DMA. And some associations with the DNA damage in DNA in tail, that is a parameter, with exposure to arsenic, we find some association and the inorganic arsenic and percentage of DMA. This analysis was performed only with the exposed group. Excuse me. This association of DNA in tail with arsenic metabolism shows that arsenic methylation has been considered, we know that it's considered a detoxification mechanism, and suggested that the methylated urinary metabolites of arsenic, monomethyl arsenious acid, or MMA, and dimethyl arsenious acid are more toxic than the molecule inorganic without methylation. And we found that this damage represented with the percentage of DMA in urine was significant, representing this damage in these people that has more elevated percentage of DMA in urine. Well, some polymorphisms that we perform in these populations are described in this table that represent the RS 374039-3 polymorphism in the population, and we found that this is not very statistically significant, so the population is in genetic equilibrium. This is the homozygote group, and this is the heterozygote group. Frequency of minor allele of the single nucleotide polymorphism of arsenic 3-methyltransferase. We can see this minor allele in our population is similar compared to San Antonio de los Cobres, but dislike or it's different compared with the Bangladesh population. So in this table, we can see the urinary metabolic profile of arsenic in relation to this SNP polymorphism. So you can see the homozygote genotype shows this percentage of related to the elevated percentage of DMA, and we found this significant difference. So that means that this profile in the relation with this polymorphism show that there could be a protective allele for the woman in this case. Finally, the ratio of genetic damage percentage of these genotypes shows that the CC allele, that is homozygote genotype, it shows a more increase of DNA damage of a percentage of DNA in tail, but there is no significant difference. Well, as preliminary conclusions of this part of the study, the women studies in these communities are highly exposed to arsenic in drinking water, which could constitute a significant health risk. Women presented low percentage of MMA and high percentage of DMA in urine. So that means that they are efficient in the elimination of this metal. And there is evidence of genotoxic damage related to exposure to arsenic inorganic in drinking water. And the CC genotype of this SNP, RS 374039-3, are associated with a high percentage of genotoxic damage compared to the other genotypes of these SNPs, but there was no statistically significant. And the CC genotype or homozygous genotype show an association with a high percentage of DMA, a low percentage of MMA in urine. So finally, I would like to acknowledge to all the people involved in this project from Sweden and from French also, and the team of the genotoxicology unit at the Genetic Institute at UMSA, and all the findings that we have for the performing these studies in this project. And also, I would like to thank to the women in this population that were participating in these studies, and thank you to all attendants here.
Thank you, Noemi. It has been an amazing talk. Thank you very much. Now we are going to make the poll, Jocelyn.
Okay.
To ask the attendants where they are from. And then they can ask their questions. They can write the questions on the chat. Also, they can ask the questions in the YouTube chat, and I can read to you, Noemi.
Yeah, I have one question from the chat of Zoom. It's Maite Berasalucci. Thank you for your presentation, Noemi. I have a question. Why were urine and blood chosen to determine the arsenic concentration and not hair and nail, for example?
Well, excuse me, I was trying to find the question to listen. It's in the chat of this Zoom.
Yeah, but it's why were urine and blood chosen to determine the arsenic concentration and not hair and nails, for example?
Okay. Well, we analyzed the urine in urine, the concentration of arsenic, because we can have the speciation there in this fluid, biological fluid, and there is a group that is working a lot of this with this exposure biomarker in Sweden. So they have all the equipments and all the procedures to analyze this arsenic exposition in urine, and also because it's most indicative of the metabolization of the arsenic in the body. But in the nails and in the hair we can find, but I think it would be not very representative of the metabolism of this metal.
We don't have more questions in the chat. Well, the poll was Bolivia. Most of our attendees of today are from Bolivia. Yeah.
Yes, Noemi, I have a question. So the lack of evidence of arsenic intoxication, like the evidence in the skin, in people in the Altiplano and also in the Puna, is related to this metabolism, the efficient metabolism of arsenic?
Yeah, yes, because the signs of this intoxication is because the metal is accumulating maybe in this in the body of the people. For example, in Bangladesh, in other places, you can see this characteristic sign of this intoxication by the arsenic. But as I show you in Argentina, in San Antonio de los Cobres, and here now in this population that we were analyzing, they have efficient elimination of the arsenic, and also the genotyping of these people show that there are, as I show you, this single nucleotide polymorphism that maybe are can protect to these people. There is genetic adaptation of these people that are living centuries of long time ago to this environment with arsenic in the water. So they have this protective genome, I can say.
Mm-hmm. And yes, so I have another question about this. So the I can't pronounce this. Arsenic methyltransferase is the name of the gene. Is it present in all the population or only in these specific populations in Diplanobuna, or also in other?
No. Yes, we can find in all the population, but in different proportion of the distribution of the homozygous, heterozygous, wild type variations, alleles, we can find differences. So if we find, for example, as I show you, this CC, that is homozygous, can be protective to the people. Also in another exposures, for example, we can find these variations of the enzymes that codify for these genes. Some are specifically for biotransformation of these environmental toxicants, but some are general. For example, glutathione S transferase is another polymorphism. Yes, polymorphisms we could find in different exposures. So there are you can find in all the populations, but in different percentages of these variations.
Okay, thank you, Nami.
So this is the population studies.
Mm-hmm.
Here there's another question.
Yeah, I have a question.
Okay.
So going on from the last question and what you said, does it look like this genetic change is something that happened slowly over a long period of time or more quickly? Do we have any idea about the evolution of this genotype?
Well, it's part of adaptation. So I don't know if I'm following your question.
Well, let me mention one other thing. I know you must have heard about the Austrian arsenic eaters, right?
Mm-hmm.
Centuries ago in Austria, they discovered that they could adapt themselves to higher and higher concentrations of arsenic, and it seemed to improve their performance and their health if they did this slowly. So I'm wondering if what they did and the change in these small group of Austrian people is similar to what happened in Bolivia, or whether it's a different type of genetic change.
Mm-hmm. Yes. Well, for example, if it's a new exposure, the genetics is the well, we born with the with the pool of genetic, each one. But if there is an adaptation of a long time, it shows that it was modified by the adaptation genetically. I don't know if you understand what I mean, but the distribution of this, or the variation of these polymorphisms could be are heritable, or we heritate of the ancestors. And if we are exposed to a new, for example, we were not adapted to something, and we cannot answer to this exposure. So this is heritated from the ancestors.
So I have another question. Is the DMA-MMA ratio a fairly good indication of a person's ability to detoxify with arsenic?
Yes. That means that the people, depending on how is the percentage of the amount of the MMA, if we have more percentage of MMA, of course, the toxicity will be presented. But if we have more percentage of DMA, it shows an efficient elimination of the arsenic. So that's how the body can detoxify for the arsenic. So it's good for the people to find this percentage. And that's what we found in this, for example, in this Uru population, that we cannot find similar data in other places. Also, compared with the Argentinian population, we found that they have also the efficient elimination of arsenic, but not in the amount of this Uru population, for example.
So you see that in the difference of the DMA-MMA ratio?
Yes.
So these women that you studied in Bolivia, do they have about the highest ratio of anybody that we know?
Yes. Yeah. It's another data interesting. And also, maybe you ask why we perform this analysis in women and not in men. It's not a discrimination. No, it's because we have different types of metabolism. So to study this biotransformation or metabolization, we decrease these another confounders, for example, with the men. And the women show a more efficient elimination of this arsenic.
We understand why that is?
Yes, it's because of this, the way of the how do you say, the endocrine system of the woman. So it's different as the men.
Hi, Noemi. We have a question from Ramiro Escalera. He says, nice presentation. And can we have some results of arsenic concentration in blood?
In blood. Have some results, but that we perform well. In Sweden, there was performed an analysis, but maybe I can get in Ramiro and I could send him some results.
Noemi,
Yeah, well, there's another, so congrats. Thank you very much for the presentation. My students and myself from South Africa enjoyed it very much and learned from it. And we have another greetings from Australia, Carolina Ahumada. Can you see questions? They ask if they can see questions in YouTube. I think you can see the video again in YouTube, but we are not going to post the questions directly.
Yeah.
no, in YouTube you have
Okay.
with greetings from people from South Africa, Mari Sanyolu, also from Oswaldo Ramos, and also from Asiahum Mudau from South Africa, and Patrick Guevara, and also Erico Freitas from Belo Horizonte, Brazil. So thank you very much to all of you for participating of this from different parts of the world.
Okay, thank you.
And I have another question, because you mentioned that the Uro population, the women in Uro, they eat more fish. And the fish is from the lake, from the Popo Lake.
Yes, it's on the Uru. Well, there was years ago there was more fish in these places, but now the Popo Lake is getting dry, so it's decreasing the consumption of fish. But years ago there was very common to eat this fish. Yes. Hi, Ramiro. How are you?
Hello, Ramiro. You have your microphone off.
Okay. It was a very nice presentation. So do you think that, well, in the southern part of Popo Lake, the populations, Cebarullo, Quillacas, etc., are quinoa producers?
Yes.
It should be interesting to assess the concentration of arsenic in quinoa, we think. Do you have some proposals in this aspect?
Yes, we have. We took several samples of these places, different kind of quinoa, and there was performed some analysis to know the concentration of arsenic, but we are not publishing this data yet. But we have this quinoa analysis of arsenic, and we found some, of course, concentration in quinoa and also in rice that they have in these places. But we know that the plantation of rice are not in this around Popo Lake, but that was a curiosity to know if there was some other of this consumption food with the concentration of arsenic. But in quinoa, yes, we have performed some.
And those concentrations in quinoa are elevated, or
yeah, yes, in some there are different ranges of concentration depending on the type of because there are very varieties of this quinoa, and of course in the places Quillacas, so there are more concentrated.
okay. I will ask you to share
Yes,
this information, please.
this information.
Okay.
Yeah.
Yeah.
Yes, of course.
We have in Quillacas treatment system, and we are
Yes.
reducing quite well this. So, do you think that this will have some impact in your studies, your kind of studies, and in a period of
Yes.
time? I don't know how many years, for example, could affect the metabolism of arsenic in the population that is consuming or treated with water.
Yes, of course, there is a need of remediation because, as I show you, there is a genotoxic damage that maybe in the a couple of years or medium exposure will be, it can become in a cancer or another a degenerative illness in these populations. So also we didn't find these typical signs of intoxication, but the people are chronically exposed, and if even they have optimal elimination or biotransformation of this metal, but that could influence in the other generation of people that could have maybe some other cancers, type of cancers, or yes, some other damage, illness because of the accumulation of this genotoxic damage. So it will be good to have these remediation activities.
Okay, thank you.
Okay, thank you.
Okay.
Hello, can I just?
Yes, Mauricio, go ahead.
Oh, yeah.
Yes, for Ramiro, questions. Ramiro, we are now submitting a proposal for better understand the relationship in between soils and some quinoa plantations, potato plantations, water irrigations, geology. So this is a new project that we are submitting to international cooperation because we want to know how is arsenic transporting from the geological origins through the soils and to the plants that are produced in this in the altiplano. If it goes well, if we have these new projects, then we will have a lot of data to understand, to better understand what is happening in this arsenic cycle. And the other thing is that we are trying to the study area is in the central Bolivian altiplano, but if we can reproduce this experiment or this assessment in South Altiplano, like in Quillacas and Cebarullo, it will be important as well. And that's all I wanted to tell.
Thank you, Mauricio.
Thank you, Mauricio. I have a question from the YouTube chat. I'm going to read it. This is Mari Sanialu from South Africa. She's asking that she would like to know if arsenic bioaccessibility test was conducted on water samples to estimate the concentration of arsenic that is available for human gastrointestinal uptake. And the other question is, how do you confirm that the concentration of arsenic in urine and blood is related to arsenic consumed from drinking water?
Yeah, okay. Well, respect to the first question, it says we didn't perform the comparison of this concentration in the blood, arsenic in blood, and only we did in urine. And she says that the concentration of, how do we know that the concentration in urine, in the water was intake in the body. I understand this, but we cannot be sure that the concentration, of course, of this arsenic intake in the water, the biodisponibility, it's another field that we have to analyze maybe. So we don't have this data of this biodisponibility of the arsenic that was introduced to the by the water. Only we have the concentrations that we found in the water and the concentration that we find in the urine that the people were eliminating as a total arsenic and adjusted by the osmolarity and some other parameters. That's that I can answer, but it's difficult to know. The biodisponibility depends on many other factors of the each people, also each person.
So if we don't have more questions, I think we are on time.
I don't see more questions.
No.
Yeah, some greetings from Mexico, from Maria Aurora Armienta. Thank you for attending. And Cecilia Echigoyen, she said that's very interesting. That's it.
Okay, thank you.
Thank you from Portugal, de Maria do Rosário Melo Costa. Okay.
Okay. Thank you very much for all the attendance. And thank you, Noemi, again. It was an amazing talk and we learned a lot today.
Okay,
really,
thank
really
you.
good talk, Naomi.
Okay, thank you. Thank you to all the attendants and to the team of this project. And see you the next webinar.
Yes, yes.
Thank you much.
It's going to be in November, but well, it's going to be me. And then in December, we have Ramiro Escalera, the last two webinars. So we hope to see you there too.
Yes. Okay, thank you.
Thank you.
Bye-bye.
Bye.
Bye-bye.
Jocelyn, can you stop the live transmission?
Yes, yes.